Sericulture is a subsidiary occupation in rural India. India has a great silkworms biodiversity, mainly because of its production of all four types of commercially exploited mulberry and non-mulberry silk. Mulberry, tussar, tropical and temperate, muga and eri. Except mulberry, other varieties of silks are generally termed as non-mulberry silks. India is the unique destination of producing all these commercial varieties of silk. India is well known for its rich biodiversity and it is recognized one of the hot spot for environment, forest, ecology, tourism in the world map. It is also recognized that a symbiotic relationship between tribals and forest, which is essential for sustainable environment and forest management. Forest Development Corporation should closely associate with tribal peoples in the protection, regeneration and development of forest, as well as to provide gainful employment to the tribals to their economic standard. Development of non-mulberry sericulture is of special relevance in northeastern states such as Jharkhand, Chhattisgarh, Bihar, Madhya Pradesh, West Bengal, Orissa, Uttar Pradesh, Assam, Jammu Kashmir and some southern states are Andhra Pradesh, Maharashtra, Karnataka, and recent observation was also noticed in Kerala suitable the exploitation of vast areas of food plants under natural forest and rise in wetlands can further increase production of tussar, muga, eri. In India alone, having more than 500 species of Saturday found to occur in different places. Wild silk moths includes tropical and oak tussar silkworm, eri and muga silkworms are reared on the wild trees but can also reared under complete human control. Most of these silkworms largely distributed in Asia, but some of them distributed other part of the world. Most of the research and development of technology is confined to Asia, particularly China, India and Japan, and many research are ongoing developing for the non-mulberry sericulture sector in this region. Based on the ecological condition, different food plant distribution, presence of ecotypes and species of diverse nature in coexistence, it is speculation that northeastern India is possible home for the origin of Antheria species. In India, both tropical and temperate tussar silkworms are found to occur. In tropical region, Antheria mylitta, commonly called as Indian tussar, is commercially reared, which mainly thrive on the food plants of Assam and Arjuna, whereas in sub-Himalayan belt temperate region, Antheria proili, which is synthetic species evolved by crossing Antheria perni and Antheria roili, is rare on the leaves of oak plants, and this has been yet to commercially exploited in large scale. Muga silkworms, Antheria assame, is largely reared by the farmers of northeastern India, particularly in Assam, whereas eri silk, product of domesticated silkworm Pilosomi racemi, that food mainly on castor leaves, which is practiced mainly in northeastern part of the state and Assam. The tussar silkworm Antheria yamamai found to occur in Japan and Antheria perni is recorded in China, which is commercially reared. The insects belongs to the family Saturnidae, distributed worldwide, particularly in many parts of India. Non-mulberry Sericulture fauna belongs to the family Saturnidae, superfamily Bombacidae, are mostly wild silk moths. These are used as important tools in basic entomological and biotechnological research in various countries in the world. These are medium to very large in size, and this family insect include the largest moth. Adult insects have a wing spring of 3 to 15 centimeter length, relatively small head and densely air bodies found to occur. Larvae are usually very fleshy, with clumps of raised bristles. Caterpillars mostly found on leaves of trees and shrubs. Some causes severe damage. Pupa develops in silken cocoons. Wild silk moths are reared on wild trees but can also be reared and breed under complete human control. They complete their life cycle of 4 different metamorphosing phases: egg, larva, pupa and adult. Of this, only larval stage is feeding period. The range of food selection of this insect is wide. Their cocoons are bigger than those of the domesticated silkworm. Out of 4 stages, the egg emphasizes the embryonic development, which lasts for about 7 to 8 days. The larval stage has 5 instar and total larval duration is about 28 days. Pupa stage remains for about a period of 7 to 25 days, followed by emergence of adult moth. The male and female moths coupled and reproduce since they are only reproductive stages. The family classification and different species are given below. Coming to scientific classification of wild silk moth: tussar, kingdom Animalia, phylum Arthropoda, class Insecta, order Lepidoptera, family Saturnidae, genus Antheria, species Antheria mylitta, Antheria assames, Antheria sivalica, Antheria royli, Antheria compta, Antheria elfri, Antheria prithvi, Antheria andaman. Coming to the classification of wild silk moth Muga, common name Muga silkworm, genus Antheria, species Antheria assama. Coming to the classification of wild silk moth Eri, common name Eri silkworm, genus Samia, species Samia cynthia saraceni. India having rich sericulture fauna and flora added to the heritage of biodiversity, particularly wild silk moths are widely available. The insect which produce tropical tussar silkworm Antheria mylitta drury, temperate tussar by Antheria royli, Muga by Antheria assame and Eri silk by Philosomia cynthia racini are abundantly distributed in tropical and subtropical deciduous and evergreen forest of India, and they exhibit wide distribution and genetic diversity under different climatic conditions and ecological niche. Non-mulberry serigenous flora and fauna also extensively available in Japan, which is rearing tussar silk by the silkworm Antheria roemamei, and Antheria species distributed in China, which is rearing of tussar silkworm Antheria perni, which is commercially reared. Antheria species also distributed in North America, which is rearing of tussar silkworm Antheria falciparum, is mostly distributed in North America but also not exploited commercially in America. Other non-mulberry commercially exploited silkworms are Antheria sivalica, Antheria royli, Antheria campta, Antheria elfri, Antheria prithvi, Antheria andaman, Antheria castema, Antheria kindred. The Indian tussar silkworm is natural fauna of tropical India, distributed in tropical and temperate region in the country. Various ecotypes Antheria species exist in different part of India. Distribution of these ecotypes is mainly based on forest type, soil type and ecological condition. The nature, food plants for feeding and suitable environmental play a vital role for the harvest the full potential of these ecotypes. Race identity in tussar silkworm is very important concept, and more studies are needed to understand these eco races in different regions. Conservation of this insect eco races along with related flora is very needed at this junction. Basically, the conservation plans should be encompasses whole spectrum of biota and activities ranging from ecosystem at macro level and at micro level. A condition of regular census on flora and fauna in collaboration with scientific institution and non-government organization helps assessing the field conditions and silk insect species which need conservation. The extensive cocoon collection, disease outbreaks predator attack. Besides, the species suitably are to be enlightened to the native rearers for their effective participation in insect conservation. Coming to Anthria mylitta drury, tropical tussar silkworm Anthria mylitta is distributed in central part of India. The commercially important tropical tussar or Indian tussar silk insect Anthria mylitta drury Lepidoptera saturnidae is having 44 ecoraces distributed along central India with varied phenotypic, physiological and behavioral characters. Some of the ecoraces of Anthria mylitta are Riley, Model, Bandara, Sukinda, Dhaba, Andhra local are being maintained in different locations in forest area, and they lacking behind scientific approach for conservation of this species in natural condition. Some wild tussar ecoraces like Riley, Chhattisgarh, Model and Jata, Orissa, Sariha and Laria, Jharkhand, Bandara, Maharashtra and Andhra Pradesh, besides domesticated Dhaba and Sukinda, are contributing for livelihoods and elevation the social economic status of around 150,000 Indian tribal families in India. The serigenous insect feeds primarily on Soria robusta sol, terminally arjuna and terminally tomentosa, besides varieties of secondary and tertiary food plants available in tropical deciduous forests of West Bengal, Jharkhand, Bihar, Orissa, Chhattisgarh, Madhya Pradesh, Uttar Pradesh, Maharashtra and Andhra. Anthria mylitta is a trivoltine tussar silkworm, rare during July-August rainy cocoon crops, September-October autumn cocoon crops and November-December winter cocoon crop season. The cocoon shapes, size and color vary from one to another depending on race and feeding habit. Coming to Anthria proili, temperate tussar silkworm Anthria proili is distributed in temperate places like sub-Himalayan belt of India, bones Jammu and Kashmir northwestern to Manipur in the northeast, comprising Himachal Pradesh, Uttar Pradesh, Nagaland, Mizoram, Meghalaya, Assam and Arunachal Pradesh. In northeastern region consists of comparatively low temperature and subtropical with excessive rainfall and short summer, whereas in climate of northwestern region is severely cold. Anthria proili exhibits bivoltine character, but at the same time it is difficult to rear second generation at the same location and the same species. This is one of the main problems of oak tussar culture, because cocoons from this second crop are used as seed during next springs. Oak tussar silkworm can be reared completely indoor in the northwestern states, but limited household. Some of the important food plants of temperate tussar culture are Quercus serrata, Quercus semiserrata, Quercus grifti, Quercus lycotricorpata, Quercus delbeta, Quercus semicarfolia, Quercus himalayan. Coming to Anthria campta, this Muga silkworm is also known as Dio or Divang species, found to occur in the wild state in the northeastern region in our country. This silkworm is mostly distributed in Assam and Meghalaya and neighboring state. Coming to Anthria frithii, this tussar silkworm was recorded both in the plains and the foothills of northeastern region of the country. Mostly this silkworm is available in different places of Meghalaya and Nagaland. Coming to Actias selina, this tussar silkworm also was recorded northeastern region in India. The cocoons of this tussar silkworm were collected different places of Meghalaya. This swallow-tailed moth by a trivoltine species has been reported from tropical moist deciduous forest and reported to occur in Sikkim, Meghalaya, Assam, West Bengal, Maharashtra and South India. Anthria atlasa, this muga is also known as a Dio muga, is the biggest of all the moth. This species was found to occur from the plains and the hills of northeastern part of the country. These silkworms were found to feed on the leaves of decollete plants. It is one of found in Orissa. This silkworms is not commercially reared in our country. Anthria assamese, the Muga silkworm Anthria assama that produce golden yellow color silk is a polyphagous semi domesticated multivoting silkworm The wild forms of this species were found mostly in India, particularly Brahmaputra basin of Assam and other adjoining areas of Nagaland, Meghalaya, Arunachal Pradesh and Uttar Pradesh. Besides northeast, this species also occur and its food plants is reported in Sikkim, Himachal Pradesh, Gujarat and also southern part of India. Many scientists were mentioned about the occurrence of this species in the natural population in this region. The commercial Muga silkworm raid the farmers of northeastern part of the country, and this silkworm production is limited to Assam. Coming to Samia cynthia racini, the airy silkworm Samia cynthia racini belongs to the order Lepidoptera, family Saturnidae, distributed in worldwide and particularly in northeastern region India. The airy silk production is an inhabited of northeastern part of the country, particularly in Assam. This moth has been imported from Assam, Meghalaya, Sikkim, Shimla and South Andaman, Theophilia religiosa. This species also distributed in northeastern Sikkim and Himachal Pradesh. This species distribution, food plants and rearing information is very less available due to not commercially exploited. Recent observation was recorded based on the availability of literature. This cocoons of this species was recorded very recent time in Manipura, Assam and Meghalaya. Cricula trifenstrata. Occurrence of this species has been reported from Assam, West Bengal, Maharashtra, central and southern part of India and abundant Andaman Islands. This species also not commercially exploited in this region. Different species of silk non-mulberry silkworms also distributed in the world. Anthrea species distributed even through worldwide but more abundant in Asian countries. Anthrea violeta, also called as Indian tussar, which is commercially rare in India, distributed central part of India. In case of oak tussar, mainly distributed in temperate region, and this silkworm is rearing and sub-Himalayan belt. Non-mulberry sericinaceous flora and fauna also extensively available in Japan, which is rearing tussar silk with the silkworm Anthrea yamamai, and Anthrea species distributed in China is rearing of tussar silk Anthrea perni, which is commercially rare. Anthrea species although distributed in America, which is rearing of tussar silkworm Anthrea falciparum, is mostly distributed in North America, but this also not exploited commercially in America. Other non-commercial exploited silkworms are Anthrea sivalica, Anthrea royli, Anthrea camta, Anthrea elfrey, Anthrea prithi, Anthrea andauman, Anthrea castnica, Anthrea kenveti. The moths belongs to the family Saturnidae, distributed worldwide. Lakhs of scientific literature are due in this wild silk moth. These silk moths are eco-friendly, some of them domesticated, some of them in wild in different localities of forest. The proper steps are required to identify the distribution of these silk moths, so that control for further damage for these silk moths are avoided. Tribal communities near the forest may be informed the availability of this silk moth in the particular place and supported for rearing techniques, so that they can better livelihood resources from this silk moth. Encourage the non-mulberry sericulture sector in the forest, not only control the deforestation but also conserve this wild silk moth. Further, there is a need for systematic studies, particularly different species of non-mulberry silkworms which are not commercially exploited.